So I'm currently employed as a wheat breeder at the Global Wheat Program at the International Maize and Wheat Improvement Center at Mexico. And today I'll be talking a lot about what we heard this morning from Dr. Raman Babu. We heard a lot about genomic selection, about QTL mapping, and I'll be talking about some of the real-time applications of these selection tools or technologies and how they help and what challenges do we face and how do we address these challenges. So let's make it an interactive session in between. If you have any questions, or you can feel free to ask us as well. So the first thing I'd like to show here is just an outline of what I'll be talking today. The first thing is the need to increase genetic gains. We just heard a wonderful talk from Susan about genetic gains and why do we need to increase genetic gains, and then a brief introduction to genomic selection and how does genomic selection fit in the framework of an applied plant breeding program, and what are some of the prospects and challenges of applying genomic selection. And last, I'll be talking about GWAS studies, genome-wide association mapping, genomic fingerprinting, and some of the work we've done on molecular tracking of alleles as well. So the first thing here is the need to increase genetic gains. We all live in a world where we want things very fast. You want noodles, 2 minutes. You want food pizza delivery, 30 minutes. You want something from Amazon or Flipkart, 2 days. And then you want something from Zomato or any food ordering application, you want it quick. But the rate of the variety turnover time or the breeding cycle, especially for crops like wheat, it can take you 5 to 6 years. So are we willing in this time where we have a lot of biotic and abiotic stresses, as we talked about the fall armyworm this morning, are we ready to wait for these varieties to be delivered fast, or are we going to wait for 5 or 6 years for these varieties? That's the question. So what can we do? And if you look at this figure here, you can see in this picture that the current annual rates of genetic gain for most cereals is around 1%. So this 1% might not be sufficient to achieve what we need to achieve to meet the global food demand. So what are we going to do to increase the production of food or crops or cereals, and how can we achieve that faster? So that's what we're going to look at today. And if you look at this equation, the classical equation for genetic gain, as Susan was showing a few minutes ago, you can see genetic gain depends on 4 different criteria here, which I'm mentioning. The first one is the heritability of the trait. So how can you increase the heritability of the trait? How do breeders try to do that? So you have to reduce environmental variation, which again was touched upon by Susan. You have to have good data. If there's noisy data, you're not going to get anything out of your data. And then there should be good replications and multiple testing, and the phenotyping has to be done with high precision. Only then the heritability of the traits can be improved. The second is the phenotypic variability in the population, or the square root of the additive genetic variance. And here it's about increasing the genetic diversity in a population, or the maintenance of genetic variation, and that has to be increased. And the next one is the intensity, how many good parents you select. So you can make crosses with many parents, or you can increase the size of the F2 population, so screen large populations so that you increase your chances of finding good performers. And the last thing is on the denominator, the length of the cycle interval. So as I said, it's about 5 to 6 years in wheat, and what can we do to reduce that generation time, or what can we do to speed up that cycle time? So I've mentioned a few things here. They are off-season nurseries, shuttle breeding, double haploids, genomic selection, and speed breeding. So this picture shows us where genomic selection comes into context here. So in order to speed up the breeding, you can use tools like genomic selection. So instead of putting out the plants in the field, we can select for good genotypes based on their genomic-estimated breeding values. So how has genomic selection been made possible? Again, the decrease in sequencing costs. So as you can see in this graph, a few years ago, there was, in 2001 at least, the cost of sequencing every megabyte was very high. But then with the advent of the next generation sequencing technologies, you can see the cost of genotyping has gone down. And now we can get whole genome markers for about $10 per sample, about 700 rupees per sample. So with this reduction in the cost of genotyping, again, as it was mentioned, the cost of phenotyping has gone high. what can we do? How can we better leverage this genomic information? So that's the question we have. And the concept of genomic selection was first introduced in 2001 and even before in 1998 as well. So it's not very new. Genomic selection is about 19 years old now. And what we do in genomic selection is we use dense genome-wide marker-based information. So most of us use SNP markers or DART markers or markers that are sparsely distributed. But in genomic selection, you can use whole genome-wide markers, very densely distributed markers, for estimating the genomic breeding values of individuals. So instead of looking at the phenotypic performance, which is the breeding value of an individual, we estimate the marker effects or the genomic values of the individuals and use that information, the genomic estimated breeding values, to select individuals. And here, the number of markers. So in these genome-wide marker platforms, you end up with millions of SNPs sometimes, and you might have only a population of, say, 300 individuals. So we encounter a problem where the number of markers are higher than the number of individuals. And in these cases, you cannot fit QTL models, you cannot fit fixed effect models, and that's where you have to go to random effects models or mixed models. And in wheat breeding, here you can see a picture of our breeding sites, or CIMMYT'S breeding sites in Toluca and Obregon. And every year, for about a month, several people we have to go about millions of plants, just walk and tag every plant and select the best plant. So this is wheat breeding done at CIMMYT. So this is laborious, expensive, and time-consuming. And the current cycle time is also very high. So what can we do to reduce this laborious work as well as deliver varieties to farmers quickly? And that's where genomic selection comes into play. And I'm going to quickly talk about what is a structure or what do we do in genomic selection. The first thing we do is train a prediction model. So we have lines that have phenotypes and genotypes, and that is called the training population. For example, let's take about 300 individuals which have both, let's say, grain yield data as well as genotype data. And we use that information to train prediction models. And then we use that to test or predict another set of individuals. So if you have 300 individuals and you're going to predict about 100 other individuals, and you calculate the accuracy, what is the real phenotype versus what is the predicted value. The correlation between the two values will give the genomic prediction accuracies. And then with the GBVs, you can get the GBs of the lines and then select the lines which have the highest genomic estimated breed values, and then make crosses and advanced generations, and then evaluate them and commercialize them. So genomic selection is all about you having a training population with good genotypes and phenotypes, and then predicting related individuals or lines that are highly related to your training population, and or you can either sparse test them in one environment and predict them in the other environment, and do this very fast and rapidly. You can do it in the greenhouse because you don't have to put the plants out in the field if you're not phenotyping. So you can save generations of, or you can save cycle time just by rapidly advancing the lines using genomic selection. And when I was asked to implement or evaluate genomic selection in the wheat breeding program at CIMMYT, these are some of the stages which I think are important. So first thing is to identify opportunities. So where do you think genomic selection can fit in the breeding program? Then the next thing is to evaluate genomic predictions in different stages and different populations, because the prediction accuracies might be different. The F2s might be different in advanced generations. In different populations, they might be different. And then the third step, the key step, it's just not about prediction accuracies and publishing something. It's about evaluating the relative advantage of GS over the current best practices. So how does GS compare to phenotypic selections? Or how does GS compare to pedigree-based selections? Or how does GS compare to high-throughput phenotyping? These are very important to evaluate before we vouch any strategy. And then if there is going to be a high relative gain compared to the other strategies, we can implement GS. And then the last part is the most exciting part. There are lots of stages where we found GS to be not very promising. And in those cases, those are wonderful research opportunities for us to explore more. And that's our approach at CIMMYT. So the first stage here is when you make crosses. Here you can select parents based on their genomic estimated breeding values. And then the second stage for us at CIMMYT, the F1s or the early generations, As you can see here, there are about millions of plants in the F1s, and in the F2s again, there are millions of plants. So the problem here, GS will have a maximum benefit in these early generations because there you have large families and you can make within family selections. But the problem here is that these populations are too large for genotyping. Just imagine going out and trying to genotype a million plants. Even in our labs, if we try to genotype 100 different plants, it's going to create a big problem if you don't have the right tools and equipment. Not everybody has enough infrastructure to run these large populations. So that's, again, a constraint for us to evaluate genomic selection in earlier generations. And the next one is the high heterozygosity. So the F2 generation is the maximum segregating generation. So when there's high heterozygosity, it's very hard to predict genotypes in the early generations. And the third one, again, is a low cost-benefit ratio. Even though we say that genotyping has become cheaper than phenotyping, at least in some developing countries like Mexico, the cost of line development, the cost of seed line fixation is still about $5.7, and the genotyping cost is still high. So unless we bring down the genotyping costs, we might not have GS add any value at those stages. And the next stage here is our head row stage. Again, at CIMMYT, there are about 70,000 head rows, and that's not feasible for genotyping. So we still use the pedigree, and we use high-throughput phenotyping at this stage. So we fly drones over these 70,000 lines, and we have high-throughput phenotyping information and also pedigree information for these lines. The next stage is a yield trial, the first stage of yield trials. So we have about 9,000 lines. And for the past 6 years, all these lines have been genotyped as part of a project that CIMMYT has with Kansas State. And these lines are used for evaluating genomic selection. And then the next stage, the second stage of yield testing. Again, there are about 1,000 lines every year, but we have several different traits evaluated at this stage, diseases and quality information. So that's, again, an ideal stage for testing genomic prediction models. And I'm just going to show you, Dr. Raman showed the diversity in Corteva's breeding germplasm in the morning, and I'm going to show what exists in CIMMYT's weed breeding program. So this is about 62,000 lines that have been genotyped over a period of 6 years. And there's vast amount. Every year the yield trials are genotyped, as I mentioned, and there's vast amount of diversity in this germplasm. And there's some amount of relatedness as well, which can serve well for the purposes of genomic prediction. And here are our results from genomic prediction. So here we can see genomic prediction accuracies within a set of lines. So let's say we have 1,000 lines. We use 800 lines to predict 200 lines. So that's what I have done. And on the x-axis, we have different traits. And can somebody just shout out, what are some of the traits where you can see high prediction accuracies? Just shout out, somebody. Grain color, yes. Stem rust, yes. Yeah. And can somebody think, why do you think grain color might have a high prediction accuracy? Why do you think grain color is well predicted? How complex of a trait is grain color? Breeders or geneticists? It's a very simple trait. There might be 2 or 3 genes actually which govern this trait. So it's very simple trait, and simple traits are always predicted well. And as you can see, stem rust again, it's a seedling. All those different letters there represent seedling resistant, again governed by few genes, oligogenic. So when your trait is of simple genetic or oligogenetic architecture, then it's predicted well. And this is a picture of what happens when you predict lines within the same nursery. And in this picture, I'm going to show what happens when you have a training set from a past historic training sets and you want to predict something for the future. And in this figure, again, can somebody shout out, what traits do you think the prediction accuracies have gone down? Grain yield? Yep. What is that? Okay, yep. Again, you can see grain yield, and those are the different environments in brackets, irrigated, early heat, drought. And as you can see from the previous figure, the values have gone down when you try to predict different environments. Grain yield is certainly a very hard, it's very challenging to predict across different years. And that's simply because of the heritability of grain yield. The heritability of grain yield across years, at least what we see for wheat and CIMMYT, is less than 0.3. So if you put the same lines in the next year, your performance is not going to be the same as in the previous year. So you have to be very careful with traits that have a low heritability. So unless the heritability of traits are high, the prediction accuracies are going to be low. And genomic selection is, again, not all about prediction accuracies. It's going to be, what do you do with these prediction accuracies? So we compared genomic selection versus phenotypic selection. So what happens if a breeder selects few sets of lines, and what happens if we use genomic selection to select the set of lines? And as you can see, the red color here shows the lines that are not selected by genomic selection as well as phenotypic selection. And the green shows lines that are selected by both these methods. The yellow shows, orange shows lines that are selected only by phenotypic selection, and the blue shows the lines which are selected only by genomic selection. So as you can see, there are always lines that phenotypic selection will select, but genomic selection will not select. So there are differences between the two tools. You can use genomic selection to discard lines that are also discarded by phenotypic selection, but it's not very reliable to always select the top performers. And as you can see here, how well can we predict the top performers using genomic selection? And from our experience, and I like that graphic there because it says the computer predicted that I would have a sandwich for lunch, but I ate a cake. Because it is very common to predict something based on known information. So you always eat sandwich and the computer will predict that you will have a sandwich. But sometimes you might have something differently and the computer will not predict that. So that's the same with genomic selection. Just you might have a rare allele, or you might have some rare transgressive segregants which are high yielding, and based on what you have already, you might not be able to get those high yielding alleles, and we have to be cautious with that in genomic selection as well. The next, what we did is to compare genomic selection with pedigree-based predictions. And as you can see here in this figure, the prediction accuracies in pedigree are in violet, and in genomic you have the prediction accuracies in orange. And these are the different stages of yield testing in Mexico, Obregon, Mexico. And as you can see, there was not much of a difference between genomic and pedigree-based prediction accuracies. Again, CIMMYT maintains a good pedigree for wheat lines, and so you don't see much difference between genomic and pedigree, and then again the yield heritability. So if the heritability of yield is only 0.6, your prediction accuracies are going to be still lower. So we all have to, again, get good data. And if there is no good data, if the heritability is very low, you're not going to get good prediction accuracies. And one of the reasons why we see this at CIMMYT, why the pedigree performs as well as genomics, is because the family sizes are very small. The stages where we test genomic selection are the advanced line stages. And the family sizes here are too small, and there's very little Mendelian sampling. So there's not much of sampling, Mendelian sampling variance here for us to exploit to predict well using markers. So pedigree performs still as better as genomics. And the next thing, what we did is to compare spectral indices from high-throughput phenotyping. So the picture here, you can see yield trials in Obregon, and they are huge. As I was telling you, it's about 10,000 plots in the first stage, and it goes on. So every year, more than 200,000 images are captured, and about 60,000 plots are scored every year. And we use both aircrafts, as you can see in the picture here, an aircraft fitted with a camera, and also drones. And if you combine the information from the spectral indices that you get from high-throughput phenotyping, if you combine the vegetation indices, and what I'm showing here is I'm just using NDVI. You can see in this figure here, the genomic is in red, NDVI is in yellow, and genomic and NDVI is in green. And what I'm showing is for the grain yield predictions for the drought-stressed and heat-stressed environments. And the first two bars show within-year predictions, and the next two bars show across-year predictions. And as you can see, genomic predictions are very similar to predicting using in high-throughput phenotyping. So if you were to spend $10 per sample versus just flying a drone and just paying whatever you have to pay for the drone, person who flies a drone, you still will find a high-throughput phenotyping a very feasible and viable option compared to using genomics, because they have similar accuracies. But again, one of the challenges with high-throughput phenotyping is the time taken to process the data. So unless you have an automated automated processing pipeline for all these 70,000 plots, and a breeder has a few days to make decisions, we still are not able to get the data at the right time for the breeder to use in making selection decisions. Again, time is a key factor to use high-throughput phenotyping data. And so we had a lot of questions. So what was happening? What is the difference between the different marker densities? Are we using the right markers when we try to predict? Because some of our accuracies were quite low. So in this figure, I'm showing you the markers in the breadweight genome, the distribution of markers in the breadweight genome. And we start with about 77,000 markers, which is an unfiltered set. And then when we filter for 70%, 50%, and 10%, you can see you come down with the number of markers. You go down, and especially you can see in the centromeric regions where you have a fewer number of markers, and the markers are more towards the telomeric ends. So we have 15,000 markers, 9,000 markers, 2,000 markers. And we wanted to do predictions using these three different sets and see if doing predictions with these different marker densities will have any difference. And as you can see here, I'm showing you within panels and across panels prediction. And the three bars here, the red markers with less than 10% missing data, which is close to 1,000. And then the yellow is about 9,000 markers, and the green ones are about 16,000 markers. And what we see consistently for all traits is no matter the number of markers is always not a critical limiting factor for genomic selection. As long as you have a crop which has high LD as wheat, a few number of markers are still good enough to get good prediction accuracies. This might be not true for all other crops which are highly outcrossing, but in wheat, this was our case. And even if you go down to 200, you still barely get a few, about 0.10 or 0.05 difference in accuracies. And few markers are still good. That's what we studied in wheat. And this is, again, another question that we asked. So what is the effect of family structures and predictions? And this was a question asked by somebody this morning about having related lines and predictions. So we did this with two different types, lines with no full sibs, no sister lines, and just one individual per cross, and then lines with two or more full sibs or sister lines. So the training population will have sister lines in the graph on the right, and on the left, the training population has completely unrelated individuals. And as you can see, the clear difference between them. So related genomic selection is all about relatedness. So if you don't have related lines in training and validation populations, obviously that's going to give us a low prediction accuracy. Again, there are other factors that should come into genomic prediction, and I believe there are physiologists in the room as well. So phenology. So if you're predicting grain yield, you just cannot take grain yield. Phenology plays a role. Lodging plays a role. And all these factors, and as Susan was mentioning, you can have some other damage, frost damage or hail damage, and all those factors have to be factored into the model. So we cannot just predict grain yield per se because it's too complex of a trait. So we looked at the correlation between grain yield, lodging, height, days to heading, days to maturity. And as you can see, and what I'm showing in the four different panels, in the vertical panels, are different years. And these correlations are not very constant across different years. In one year, you can have a high correlation with days to heading, and the other year, you can have completely no correlation. One year, your lines could have natural lodging. The other year, they will not have lodging. So this dynamics is very different, and we have to factor in all these dynamics in the prediction equations to be able to predict well. So a team at CIMMYT, led by Dr. Jose Crasa, is doing a lot of trying out a lot of different models, new models for using all these high-throughput data points, because the number of data points is huge. So one of the approaches that they use is the item-based collaborating filtering approach. So if you go to websites like Amazon or Flipkart, you visit the website once, and after some time you get some images telling you if you would like to purchase the item. So there is some approaches going on. There's some machine learning, there's some artificial intelligence done where they try to give you options, suggest you something if you would like to buy that in the future. So one of those approaches is the item-based collaborative filtering. So what do you prefer as a customer? So this is also used in Amazon, Netflix, and other e-commerce industries. So we've used this approach to predict. So if you have a specific heading date, if you have a specific days to maturity, and if you have a specific height, what is your grain yield going to be? So we've trained models using the IBCF approach, and in this figure you can see on the right we have lines using the IBCF approach, and when we factor all these different traits that might affect grain yield or correlated traits, you can see a real improvement in accuracy. Although that's not very significant, we still have improvements in accuracies using these traits. And some of the last things I want to talk about. So what are the prospects for implementing GS at CIMMYT? So I've touched upon different areas so far where there are challenges and predictions. And so this slide shows us what are we going to test for the future. So one of our things that we have not done so far is using genomic selection or GEBVs to predict the parents that are used in crossing. So that information becomes very crucial to a breeder who now just uses phenotypic information. So in the future, we will be having GEBVs also incorporated so the breeder has all the information to make parent selections. And then there are lots of speed breeding schemes, and so combine them. So test a conventional breeding scheme, test GS and speed breeding, and see what do you lose or what do you gain. And then in the as I've shown before, in the early generation selections, again, we have to check cheaper marker platforms, and then exist many platforms, $5, $1. We have to check some of those platforms, and also check what is the role of within and across family variants, because we have huge populations, and there's always a lot of research that has to be done on dissecting out within and across family variants. And one thing we've done last year is scaling up selections to early generations. So in this figure, I'm showing quality traits, which are always well predicted in wheat. And we've used about 3,000 lines, some of our historic training sets, to predict 8,000 lines that were not phenotyped for quality. So to have wheat phenotyped for quality for several different quality traits, the cost is about $60, which is about 4,200 rupees for one sample. So that's too expensive. Quality is an expensive trait. So we've used our training sets to predict that, and as you can see here, our accuracy is not too bad. So some of the traits like alveograph, the accuracy is about 0.6. So quality always shows promise for genomic selection, and if you look at the cost savings, instead of phenotyping 10,000 lines, you can phenotype just, you can just use training sets and predict them, and you're going to still save about $600,000. And the last thing I have is envisioning genomic selection for wheat breeding. And in this figure, I'm showing you the different traits and the correlations between different traits. So one trait might have a very low correlation with another trait, and predicting one trait is not always going to help the breeder. So breeding is not all about single traits, and we always have to incorporate selection indices with multiple traits, and we need a broader vision for implementing GS in wheat. And then since my time is up, I'm just going to skip that. And with the last two slides, we've also done a large-scale genome-wide association mapping for identifying different regions. So we have about 4,000 lines that we used, and for phenotyping different traits, grain yield in different countries in Bangladesh, Canada, Egypt. And we can see some consistent regions across these different countries. And we also put all these consistent markers along with the known genes into the reference genome of wheat. And we've also fingerprinted CIMMYT'S germplasm. So about 44,000 lines. We've fingerprinted all these lines for the grain yield alleles. So a green allele here is an allele that increases grain yield, and a red allele is an allele that decreases grain yield. So we have fingerprinted all our germplasm, 44,000 lines, and you can see breeders have pretty much done a good job in increasing the alleles of frequencies of the favorable alleles even without markers. And this is also just division of the molecular tracking of these alleles by different chromosomes. And as you can see, some of these alleles, this is over 15 years of wheat breeding at CIMMYT, and some of these alleles have really come to high frequencies just by a breeder selection. And we can do much more with the genomic tools that we have, and that's what we are trying to achieve. And all of this work has just been published last month in this paper in Nature Genetics, and if anybody is interested, yeah, it's available, and yeah, you can read it and it'll help you. And with that, I would like to thank everybody involved in this, CIMMYT, a lot of funders, and I would like to thank all the organizers for giving me this opportunity. And I'm also very grateful to all my professors who taught me a lot of these fundamental concepts which I apply today in all this work that I've described. Thank you very much.
The session is now open for discussion.
Hello, ma'am. Myself, Shaukat. So actually, my question is, you told you can employ genomic selection at different stages. So there, at different stages, how can you decide training population and a test population? What criteria you will set for the selection of the training and breeding population?
The criteria is just they should be highly related. So you have a set of lines, you should either have sister lines or very highly related lines in the training population so that you can predict value validation population. If you're selecting completely unrelated lines as your training population, it is not, genomic selection might not give you high accuracies.
Okay, and then ma'am, for different traits, you told the GBV values. For grain yield, some values there, and for some other traits, some values there.
Yeah.
Cannot we tell for 2 or 3 traits or 4 or 5 traits at a time?
Yeah.
The value is only one. This is a best plant or a best genotype.
Yeah,
Like
sure.
that, can we say?
Sure, yeah, that's a good question. So you can get the GEPVs for different traits, and the breeder can use those different GEPVs to make his selection decisions. So some of the accuracies may be high, some of them are low. What we are doing currently is integrating them with phenotypic values also. So we have not entirely done genomic selection per se. So we've always used the phenotype. Also, we have used the GEPVs to make selection decisions. So you can have multiple traits and also use a selection index or something to select multiple traits. Yes.
Okay. Thank you, ma'am.
Thank you.
Thank you, ma'am, for sharing your thoughts on the strategies for increasing the genetic gain. May I call upon Dr. S. Geeta, Director, CPPG, to honor the guest speaker.
I just wanted to make one quick comment to Philomin and then one quick question to Philomin as well. I think it was very, very well presented, Philomin. First of all, I would like to congratulate. Genomic selection for the benefit of students is becoming a kind of a mainstream breeding approach. I think it's really important that you take note of this presentation. There were a lot of many positive, actually very empirical evidences for how genomic selection can be implemented in the breeding program. If you're really aspiring for a career in seed industry or in any of the even public sector institutions in the years forward, it is really, really important that we learn this particular approach very well. So if you have an opportunity to pursue research in this area, please push your chairman and your teachers to see if you have an opportunity to do research in this area. That is the only way that you can learn more on this. And one possible constraint in universities is that there may be lack of resources for generating this kind of information. And we are really, really fortunate in India to have so many other international institutions. We have CIMMYT, we have IRI, we have ICRISAT, we have so many private sector organizations who are actually not knowing what to do with that kind of information. They have generated so much information, everything is available with them, and they are just waiting for some collaboration, for some request from the universities or from other public institutions. So please push. You learn what's happening outside the world, outside the university, and please push your faculty, your chairman to have some collaboration so that you can get access to that kind of data and you can really make use of that kind of data in the breeding program. This is a wonderful opportunity. This is one way of increasing the employability of plant breeding, molecular breeding, as well as biotechnology graduates. So please take note of that one. And one quick question to Philomin. So Philomin, what's happening on the imputation side in CIMMYT? So what I learn is that a lot of high density genotyping is happening. Are there any gains made in terms of imputation? Because what we get to understand is that most of the crosses are biparental pedigree crosses. Yep. So just by improving our imputation methodologies, we should be able to fill in a lot of information, and that can be used in
Yeah.
genomic selection?
Sure, absolutely. So that's a very good question. And some of our major parents, like whole log 100, has been used in like 5-10% of all our crosses. So what we've done is we'll be doing whole genome sequencing of those big, widely used parents. So that's going to be part of a project at Kansas State and USAID. So we'll be doing whole genome genotyping of those, and then we'll be imputing the other lines using those parents. So we've selected about 200 parents, and that's underway to do a high density genotyping of those parents and imputing the other ones. Yeah. Thank you.
In line with the program schedule, we have the poster presentations to be conducted soon. The participants are requested to be in the allocated slots and to wait until further instructions. Everybody are requested to assemble downstairs for the poster presentations. Please reassemble in the hall after the poster presentations for further events. Please reassemble at 6:45 p.m. for the further events.
Sir, and the image is sir. This is a backdrop of open. Okay. Gmail.
Nanotech at tna.ac.in.
Nanotech
At tnau. tnau. tnau.ac.in. Just 1 hour. Yeah, 5:30 to 6:30. Dean PGS will be Dean PGS on that. You open my team, download download
PDF sir.
Varado.
Image or நான் பேங்க் ஸ்க்ரீன் சேவராக வச்சுட்டேன். வேணா அதை எடுத்து விட்டுட்டு சார் இல்லை சார் அது பிடிஎஃபாக இருக்குங்காட்டிக்கு இப்படி வியூ ஆகுது கீழே இருக்குது சார்.